Hello. Today what I'm going to do is just narrate skeletal muscle contraction for you. I'm going to take it really from activation all the way to excitation, contraction, coupling, which will then provoke cross-burst cycling. So, like we've done in the past, I've made kind of a series of diagrams here for you. So, where we're going to start in this conversation is really kind of at what you could call the motor unit. So, or what you would absolutely call a motor unit. So I've made a little drawing where I have a cross section of the spinal cord. This would be the ventral side here, and this would be the dorsal side, and I've drawn in a lower motor neuron for you. I have the ventral horn gray matter right here. This is the ventral root leading to the mixed spinal nerve, and then I've kind of edited this and kind of drew an arrow here, basically kind of breaking it, saying, hey, this goes a long way before then it finally gets to individual muscle fibers. Now, what I should do or could do here would be to draw multiple muscle fibers. In fact, let me just do that. Let's just augment this just a little bit and indicate here that this lower motor neuron is not just going to one muscle fiber, but it is going to branch out, and it's going to go to, in this case, several muscle fibers. Now that could be as many as 4 or 5, or it could be hundreds of muscle fibers, and that kind of leads to the discussion of small and large motor units. So I've kind of added some extra fibers here that are going to be stimulated by this one lower motor neuron. Now, let's follow that stimulation. So let's assume an action potential depolarizes down this lower motor neuron. And so as the action potential depolarizes down that lower motor neuron, there's that wave of sodium influx followed by the repolarization wave of potassium efflux. At this point, that should be a really familiar thought and conversation that you can have. That action potential reaches the axon terminal here. So we have the axon terminal, and we have one skeletal muscle fiber here indicated. And we can draw that action potential again depolarizing down toward the axon terminal. And you probably at this point remember that calcium is the last ion that influxes into the axon terminal and will provoke the release of neurotransmitter from the axon terminal. So that's what we've indicated here, is the release of neurotransmitter from vesicles in the terminal region of the axon, or the axon terminal. In this case, of course, we're looking at a somatic motor neuron, so the neurotransmitter being released is acetylcholine. And we're showing it here. This whole unit would be called the NMJ, which stands for neuromuscular junction. And those little dots of yellow represent acetylcholine crossing the synaptic cleft and then binding to these receptors. And those receptors will be nicotinic acetylcholine receptors, also called cholinergic acetylcholine receptors. And when acetylcholine binds to those receptors, they open up and they allow the cation sodium to influx into the skeletal muscle fiber. And what that does is it creates a graded potential. And if that graded potential is strong enough to reach threshold, the result will be an action potential being propagated along the sarcolemma. And sarcolemma, let me draw that out for you, write that out. Along the sarcolemma is the word for the plasma membrane of a skeletal muscle fiber. And it also, that action potential doesn't just stay out on the surface or out on the sarcolemma, but it dives into the muscle cell via structures called the T tubules. That first T stands for transverse tubules. And that drives the action potential not only on the surface, but into the interior of the skeletal muscle fiber. From that point, that action potential depolarizing along the sarcolemma and into the muscle cell via the T tubules causes the sarcoplasmic reticulum, or the SR, to release calcium. And what I've depicted here, we're zooming in again. So let me just be clear, we're zooming in. And we're looking at one myofibril. You'll remember that myofibrils are the cylinders that are packed inside of the skeletal muscle fibers. And the myofibril, we see the sarcomere-like structure from Z disc to Z disc. We see thick and thin myofilaments. At the myofibril, the sarcoplasmic reticulum is releasing calcium, and that calcium has the action of binding to a regulatory protein. It binds to a regulatory protein called troponin or troponin complex. And I've kind of indicated that like we have in the text in a yellow color. So that represents troponin. Here troponin is bound with calcium. And as troponin is bound with calcium, it changes its shape. It releases the long filamentous protein, tropomyosin, from the active binding sites on actin. And what is allowed to happen at this point is what is called the Crossbridge cycle. So these myosin heads, they will bind to actin. That's also referred to as they form a crossbridge. The action is crossbridge attachment. The second thing that they do is that they have a working stroke or a power stroke. I call that rowing. The next thing that happens is that they detach, and detachment requires the attachment of ATP to the myosin head. And then after they detach, ATP is hydrolyzed. It's broken up into ADP, adenosine diphosphate, and an inorganic phosphate molecule. And the energy that is liberated because of the breaking of that high energy phosphate bond is used to complete the fourth step, which is the re-energization or the recocking of the myosin head. And that will continue to happen. This will continue to happen as long as there is calcium present. In other words, calcium is still being released from the sarcoplasmic reticulum or has not yet been resequestered into the sarcoplasmic reticulum. And the other requirement is adequate ATP. And so the Crossbridge cycle continues really as long as the muscle fiber continues to be stimulated with acetylcholine at the neuromuscular junction. So that will prompt continued crossbridge cycling. As crossbridge cycling occurs, what happens is that the length of the sarcomere shortens. So we get shortening of the sarcomere. The two Z discs, they get pulled closer to one another. As the sarcomere shortens, the myofibrils shorten. As the myofibrils shorten, the whole skeletal muscle fiber shortens. And as muscle fibers or muscle cells within a skeletal muscle shorten, then the whole muscle itself will shorten. And tension is built, and that is called muscle contraction. So I hope this is a good review for you of the events from activation to excitation to crossbridge cycling. Have a great day.